Greetings, everyone, and welcome to this edition of Medicine Grand Rounds. It's 12:01, and so we'll go ahead and get started. I'm Conrad Lyles. I'm the Associate Chair in the Department of Medicine, and I have the honor of introducing our two speakers for this Medicine Grand Rounds. And we're honored today delighted to have our Chair of Medicine Scholars Award recipient for 2022, and then also our Diversity Academic Development Scholar Award recipient, or DADS Award recipient, also speaking for 2022. And our first speaker will be Dr. Chang Li. And Dr. Li is a research assistant professor in the Division of Medical Genetics. And the Chair of Medicine Scholars Award is provided, or has been, or we have developed it to help the transition from a senior fellow or acting instructor level to a assistant professor level, and the transition from sort of the K funding up to an R01 type funding. And today, Dr. Li is our recipient, and he will talk to us today about in vivo precision genome editing for β-hemoglobinopathies. And he will give about a 20-minute presentation. Or 25-minute presentation. If you have questions that you would like to ask, please put them in the chat, and then I'll pose those questions to Dr. Li before we go on to the second presentation. So, Dr. Li, I turn it over to you.
Okay. Good afternoon, everyone. Thank you, Dr. Lyles, for the introduction, and also thank you and the review committee for giving me this award. Let me move this a little bit downward. Okay. So this is my disclosure. I don't have a conflict of interest to disclose. About me, I received my PhD from the University of Chinese Academy of Sciences in the end of 2014, and studied I'm sorry, there's a little issue with my computer. Let me say. All right, I studied HIV entry mechanisms and the gene therapy for HIV. So then I had a postdoc at the University of Maryland. In 2016, I moved to UW, studying in vivo hematopoietic stem cell gene therapy. So my postdoc mentor is Dr. Andrew Lieber. I am currently a research assistant professor in the Division of Medical Genetics. My research interests are the development of cost-effective gene therapy strategies by precision genome engineering for blood disease, with a focus on β-hemoglobinopathies. So globally, there are over 400 million carriers of abnormal hemoglobin genetic traits. So most common in resource-limited regions. HSC gene therapy is a very promising treatment approach because it uses patient HSCs and therefore not dependent on stem cell donation and donor availability. So currently there are three major strategies: gene addition of a functional beta or gamma globin gene, which is expressed in fetus and normally silenced soon after birth. So reactivation of fetal hemoglobin and repair of the sickle mutation. Conventional HSC gene therapy is based on a ex vivo strategy involving HSC connection, engineering and culture in vitro in test tubes, and then infusion back to the patient after myeloablative conditioning. So it can be used for a number of blood genetic disease. So despite encouraging progress, a number of potential side effects are associated with this process. So in other words, it's a complex, risky, and very expensive procedure. Just as give as an example, Zynteglo, approved by FDA for the treatment of beta thalassemia. The cost is $2.8 million per patient, making it the most expensive medication in the country. We are developing a simplified HSC gene therapy strategy involving only subcutaneous and intravenous injections It is performed by mobilization of HSCs from bone marrow into peripheral blood, followed by injection of viral vectors expressing transgenes. The mobilized HSCs are transduced in circulation and then home back to the bone marrow shortly after this procedure and persisted there long term. This approach requires no myeloablation, no transplantation, and no ex vivo HSC culture. The efficacy and the safety of this approach has been tested in a number of animal models. Potential limitations associated with IV injection of viral vectors can be efficiently addressed by pharmacological interventions and optimized vector platforms. Our current protocol uses a chimeric form of help-dependent adenoviral vector with a number of favorable features, including all viral-coded genes removed, efficient HSC transduction by utilizing CD46 as a receptor, up to 35 kb packaging capacity, and different design for episomal and integrated vector status. Using this in vivo HSC transduction strategy, we delivered a CRISPR system targeting the BCL11A eraser binding site in the HBG promoter for reactivation of fetal hemoglobin. The vector also has a MGMT cassette, which provides a mechanism for drug resistance and selective expansion of gene-modified cells. The experiment was performed in beta-yak CD46 mice, which resemble human fetal hemoglobin expression. After in vivo transduction and selection, we measured a significant increase of gamma globin in red blood cells. As we know, CRISPR makes double-strand DNA breaks because there are two HBG genes, HBG1 and 2. CRISPR cutting at these two identical targeting sites may result in deletion of the fragment in between. So indeed, we found significant intergenic deletion indicated by this shortened PCR applicant using primers to show above these red arrows. Hans-Peter Kim's group found that CD34 cells from non-human primates bearing the intergenic deletions are rapidly lost following transplantation, indicating loss of engraftment capability. So another strategy we used is targeted integration of a functional gamma globin gene into the AVS1 site, which is considered a safe genomic harbor and supports high transgene expression. So after in vivo transduction, we measured the gamma globin production level reached 25% of mouse beta major group protein, which could be therapeutic in clinical setting. So it is generally accepted that 20% is the threshold required for providing therapeutic benefits. So however, targeted integration mediated by homology-directed repair is an inefficient process. As we can see, not all the animals responded. Besides, HDR is also dependent on double-strand DNA breaks. So we found a significant toxicity in human CD34 cells caused by CRISPR system, shown by this compromised engraftment in humanized mice after CRISPR vector transduction. Although this can be partially prevented by blocking CRISPR activity using anti-Cas9 peptides. Double-strand DNA breaks cause other adverse effects, such as DNA damage response and the large fragment deletion around the targeting site. So therefore, we turn to more precise editing tools. Base editors consist of catalytically deactivated Cas9 linked with a deaminase, and sometimes with other components that can improve its activity. So there are two main types of base editors, cytidine base editors and adenine base editors, mediating C2T and A2G conversions, respectively. They are independent of double-stranded DNA breaks and mediate more precise genomic edits than conventional CRISPR system. So we aim to create naturally existing mutations in the HBG promoter region, so associated with a higher fetal hemoglobin expression. also called HPFH mutations, the hereditary persistence of fetal hemoglobin. So beta thalassemia or sickle cell patients with these mutations usually show mild disease symptoms, demonstrating protective effect of these SNPs. Specifically, by screening, we identified a guide RNA that can create the minus 113 A2G base conversion and therefore activate gamma globin expression. So we constructed vectors expressing this guide RNA and a ABATE base editor as well as a MGMT cassette. So we then performed a study in thalassemia patient CD34 cells from 3 different donors. So after transduction with the base editor and extra setting with selection was performed. So followed by in vitro erythroid differentiation. We measured the 25% target site editing. With editing, the editing level was raised to 55% at day 18 after differentiation, and also therapeutic gamma globin production. The edited cells show much lower reactive oxygen species and improved erythropoiesis with more mature erythroid cells after differentiation. So demonstrating significant phenotypic improvement. So importantly, we focused our studies on in vivo HSC transduction after HSC mobilization with GCSF and MD3100. So beta-YAC CD46 mice were intravenously injected with the ABE vector. Here we used an early selection approach by utilizing the episomally expressed MGMT in this vector. So no transgene integration here. The mice were followed for 16 weeks, and then a second transplantation was performed and monitored for another 16 weeks. So at week 16 after transduction, we've measured around 60% target-based conversion in the primary mice. And this level remained stable in different lineage cells as well as in lineage minor cells, which contain the hematopoietic stem cell fraction. So demonstrating editing occurred in long-term repopulating stem cells. So since base editors are not dependent on double-strand DNA breaks, so we measured much lower level of indels, around 1% at week 16 in the primary mice. And this is around 50-fold lower than the desired base conversion rate. So only minimal level of intergenic deletion was measured compared to the CRISPR system here. Since the base editing strategy does not remove the sickling beta globin gene, which can compete with the reactivated gamma globin for the alpha globin binding. So the ideal strategy is to directly fix the root cause of sickle cell disease. We aim to achieve this by in vivo precision prime editing. Prime editors consist of a catalytically impaired Cas9 linked with a reverse transcriptase. So also include a kind of extended PEG RNA, this green line here, called PEG RNA. It can mediate templated target DNA synthesis via reverse transcription. Prime editing is independent of double-strand DNA breaks and is capable of generating all types of genomic modifications. So as we all know, sickle cell disease is caused by one single nucleotide mutation in the beta globin gene, leading to hemoglobin polymerization and the sickling of red blood cells, as shown in this sickle cell disease patient blood smell. So by collaborating with David Liu's group at Harvard, so who invented the prime editing, we designed a HD80 vector expressing P5 max, which is a latest version of a prime editor. All components over 17 kb in length delivered in one vector. The expected editing is to convert GTG, this sickle codon, into a GAA sequence. So apparently, the T2A conversion is to fix the sickle mutation, while the G2A conversion at the third position is to prevent the PE from continuous editing by destroying the POM sequence, which is required for guide RNA binding. Here we performed experiments in sickle cell disease patient CD34 cells. To raise the editing, we conducted one round selection after transduction with the P5 max vector, so followed by in vitro erythroid differentiation. At the end of differentiation, we measured 71% T2A conversion, and a significant phenotypic improvement was found in the edited cells, reflected by reduced reactive oxygen species and improved erythropoiesis. So the cell smells show more matured erythroid cells after prime editing. We used the Towns mouse model for animal studies. It is a murine model for sickle cell disease. We crossed Towns mice with our CD46 transgenic mice to facilitate virus transduction. As you can see, these mice have a higher level reticulocytes, nucleocytosis, like abnormal hematocrit and insufficient sickling red blood cells in an in vitro sickling assay, and also enlarged this gigantic spleen caused by extramedullary hematopoiesis. So next, we conducted an ex vivo HSC gene therapy study using lineage minor cells from CD46 Towns mice. Lean minor cells were transduced with P5 max vector and infused into irradiated B6 mice, followed for 16 weeks, and then followed by another secondary transplantation. The engraftment was near complete. The editing level was more than 95% at week 16 in the primary mice. Note that this is without any selection. So next, we focused our study on in vivo transduction with a similar earlier selection approach. The mice were followed for 16 weeks, and then a secondary transplantation was performed. Editing level at the targeted site was around 40% at week 16 after transduction. So this level remained stable in different lineage cells and the lineage minor cells in the secondary mice. Colonial analysis showed distribution of editing in HSPCs, stem cell and the progenitor cells of full mice. An average of 20% of colonies have biallelic editing, and 40% colonies have at least one copy of edited gene. So we analyzed the hemoglobin composition after treatment. Using isoelectric focusing, we found around 50% sickling hemoglobin S in vivo transduced animals were replaced with a normal HbA. In ex vivo treated mice, the replacement was near complete. We also examined the globin chain composition by mass spectrometry, which can well separate the two forms of beta globin, and observed a similar level of correction. Then we performed the systematic phenotypic analysis in the treated mice. So these mice show near complete or largely reduced percentage of sickling cells compared to untreated cells, and also near normal percentage of reticulocytes. So normalization of hematocrit and nucleocytosis was shown by blood analysis, and the reduction of spleen size was also observed in the treated mice compared to naive Towns mice. To briefly summarize, our vectorized base editor generated therapeutic base editing in thalassemia patient CD34 cells. In the beta-YAC CD46 mice, in vivo HSC transduction led to over 60% editing at the -11A2G site, and 25% gamma globin of human beta globin expressed in erythrocytes. In a sickle cell disease mouse model, in vivo HSC primer editing with the P5 max vector achieved an average 50% of HBS replaced by HBA. So this is resulted from 40% edited alleles in HSPCs with 20% biallelic edits. So these numbers compare favorably with data from ongoing ex vivo HSC gene therapy trials involving lentiviral vector-mediated gene addition of anti-sickling globin genes. So our approach involves a single intravenous injection of an all-in-one episomal HDAD vector after HSC mobilization. So no integration here. The treated mice show greatly improved SCD phenotype. Future directions. So while we're working on strategies to further improve the efficacy and the safety of the in vivo approach, including more selective and compatible mobilization regimes, which is important for sickle cell disease patients. Alternative selection approaches and more efficient editors and vector platforms, such as ABATE to create the non-pathogenic Marcasa variant, and HDAD vector based on novel serotypes, and also test in large animal models, which are more similar to humans. I'd like to thank my postdoc mentor, Dr. Andrew Lieber, who is always very supportive for both my career and research, and also Lieber Lab members. I also like to acknowledge our collaborators, Dr. Tess Peter Kim, Dr. Daniel Papianopoulou, David Liu from Harvard, and Dr. Yanaki and Aphrodite. They performed experiments in patient CD34 cells. So this is a photo of my little family. My second daughter was born like right before the pandemic. She's almost 3 now. Without their great support and understanding, it's impossible for me to do my research. Thank you so much for your attention, and I'll be happy to take your questions. Thank you.
Thank you, Dr. Li. In terms of questions for the audience, we can't really call on you to in this webinar format, so please put your questions in the chat, and then I will pose them to Chang. But while we're waiting on the questions from the audience, because there are 2 people who had raised their hands, which we can't really call on them, but put your questions in the chat, and I will ask them of Dr. Li. Chang, of these 2 strategies to treat beta thalassemia and to treat sickle cell disease, which ones do you think is most promising to take to prime time? In other words, to translation into humans?
So currently, people are more focusing on the gene addition strategy using a lentiviral vector to put a healthy copy of gamma or beta globin, anti-sickling beta globin. The prime editing strategy is very it's a very recent technology. I think it's very promising, but the thing is, it needs to be comprehensively tested, especially in clinical settings. So this is I think this is our study is the first one to try this. So it's a lot to be done, but I think it's very promising, the prime editing one, because it fixed the root cause.
Yep, and like I'm waiting. If any audience members have any questions, please put them in the chat. We don't really have the capability to call on you to ask your questions orally in this webinar type presentation. But I agree with you, Chang. I think that the sickle cell type of editing project looks like the one that could be pushed along more quickly to certainly to clinical trials and perhaps then onto patient populations. I agree with that.
Yeah. A lot is going on, the clinical trials.
Any other questions then? Please put them in the chat and I will pose them to Dr. Li. I don't see any others. And you said also that you're going to try to optimize mobilization strategies for the stem cells so they could be transduced. Besides GCSF and AMD 3100, what were the other strategies that you were going to investigate?
So I'm sorry, I caught this. I'm having this like nasal voice. Yeah, so a couple of drugs we are interested in testing. One is it's called the group beta. I didn't read it out. So the other one is W106. So we tested this group beta like in a previous study in a thalassemia mouse model. So and we plan to test these two drugs in the Towns sickle cell disease model. So these are the two primary targets. Probably there are more. Yeah, we'll definitely look into that.
Okay. Any other questions from the audience? Please put it in the chat. But if not, Dr. Li, thank you. Nice work, interesting presentation. I hope things go well and you continue to make fine success in your research. And
thank you so much.
We're very proud of your accomplishments on behalf of the Department of Medicine. I want to thank you for your accomplishments and for doing some very interesting research and major problems of public health importance in certain regions of the world. Thank you very much.
Thank you for your support, Dr. Lyons. Thank you.
And our second presentation for this grand rounds will be our DADSA Award winner for 2022. And the DADSA Awards were established to promote the transition from senior fellow to acting instructor, or more the transition from being a fellow to obtaining a K award. And this was designed for individuals that were interested in doing projects or research to meet our goals of DEI in the Department of Medicine. And I'm proud to introduce our award winner, DADSA award winner for this year, which is Dr. Busola. Oluwole. I think I got it right. I had it right before, but Dr. Oluwole is also going to be talking about some hematology. He's going to be talking about sickle cell disease. She is currently an acting instructor in hematology here at the Department of Medicine, and also has activities over at Fred Hutch. And she's going to be talking to us today about neurocognitive functioning of patients with sickle cell disease. And once again, if you do have questions, please put them in the chat, and I will pose them to Dr. Oluwole at the end of her presentation. So, Dr. Oluwole.
All right, afternoon, everyone. As mentioned, my name is Busola, and today I'm going to be giving a talk about neurocognitive functioning, imaging assessment, and association with anemia in adults with sickle cell disease. I have no disclosures. So a little bit about myself. There I am at a young age. So I grew up in Nigeria, and at the age of about 11 years old, my family and we moved to Bay Area, California. Since then, in terms of my journey, I completed medical school, and also as part of the clinical scientist training program, also completed a Master of Science in Clinical Research at the University of Pittsburgh. I stayed there for residency as part of the global health track. And then in 2019, came here for a hematology fellowship. In terms of my clinical interest, it's really in sickle cell disease. And my research focus is on neurobiological complications in patients with sickle cell disease. And so sickle cell disease is an inherited genetic blood disorder. It's caused by a point mutation in the beta-globin gene, and there are different subtypes. The most common one and most severe form is the homozygous S sickle cell anemia, which we call sickle cell anemia. We also have other sickle cell subtypes that are associated with co-inheritance with other hemoglobinopathies, such as beta thalassemia. The hallmark of the disease are really these very painful vaso-occlusive crises. In the United States, about 100,000 Americans are affected, and it's considered a rare disease in the US. However, worldwide, there are about 300,000 babies, newborns are born yearly. Most of them are in sub-Saharan Africa, where most of the population is actually in Nigeria. About over 50% of the population are in Nigeria. So regarding the pathophysiology of sickle cell disease, it's very, very complex. This is sort of a simplified model that shows in sickle cell disease, we get in the state of stress or hypoxia, there's polymerization of hemoglobin S that leads to formation of these sickled red blood cells that you see here. And this can cause occlusion of microvascular occlusion and lead to vaso-occlusive crisis and some other organ damage as well. We also have, as a result of these unstable red blood cells, there's chronic hemolysis, there's anemia that also occurs. And so the way I think of sickle cell disease is that it's really a disease that can impact any part of the body. There are some acute complications that arise from sickle cell disease, and then there are some chronic complications as well. The part of the body that I'm especially interested in at this time is the brain. Neurovascular complications are unfortunately common in sickle cell disease. You have overt strokes, which are strokes with apparent neurological deficits. You also have silent cerebral infarcts, which are really lesions that we see, structural lesions that we see on imaging without any clinically apparent neurologic deficit. And then we have cognitive impairment. When you compare patients, children who have these silent infarcts to those who do not, there is really a 7.2 decrease in full-scale IQ. And then when you also compare those who have overt strokes to those with the silent infarcts, those with overt strokes have about 11-point decrease in full-scale IQ. However, what's also interesting is that even in the absence of strokes, we still see cognitive impairment. Cognitive impairment, as patients are living longer, it's especially important to try to understand the development of the etiology and the pathophysiology leading to cognitive impairment outside of strokes. Cognitive impairment can impact employment, education, adherence to medications. And really in the past decade, or a decade or 2 decades, there have been really emergent data focusing more on cognitive impairment in this population. Associated factors have been shown to be maternal education level, nutritional status has also been shown to be associated, socioeconomic status, but there's still really a lot we need to know and understand about the development of cognitive deficits. A lot of the prior studies have been done in the pediatric population with very limited studies in adults. Most of them have been cross-sectional, and so we don't really have that much data longitudinally. And also the association between cognitive impairment and functional abnormalities is also limited. Most of the studies have looked at structural abnormalities, though we do have some studies looking at functional abnormalities, is still limited. And then there's also varied results regarding the impact of anemia. And this is a figure that I got from Dr. Jordan and Dr. Dubon, which really highlights what we know about the pathophysiology of neurological injury in sickle cell. So what we know in sickle cell anemia, in sickle cell disease, there's lower hemoglobin. Patients are more anemic than when you compare to healthy participants on the left here. Also in sickle cell disease, has a compensatory mechanism for decreased hemoglobin, decreased arterial oxygen content. You have increased cerebral blood flow. Also, when you look at cortical vascular reactivity, which is really vascular response to try and increase cerebral blood volume, it actually is less when you compare to healthy participants. And so putting all this together, my aim for my current study is really to look at the association between anemia and cognitive functioning, and also to look at other imaging modalities other than our conventional MRI. So the central hypothesis is that patients who have higher hemoglobin levels will have better cognitive performance than patients with lower hemoglobin levels. And this is in adults with sickle cell disease. So the first aim will really to look at baseline cognitive functioning, look at baseline functional MRI and correlate these with hemoglobin levels in adults with sickle cell disease. And then the other part of the study is really to have a longitudinal analysis. So looking over the period of 2 years, if we are able, what we see regarding the association with of cognitive functioning and hemoglobin levels, especially as patients are going to be starting on treatment that improves anemia. So this is the methodology. This is a prospective study conducted over the course of 2 years. Participants, really adults who are over the age of 18 with any diagnosis of sickle cell disease, any subtype. And at the bottom here is really the study schema for this study. So we get baseline imaging, baseline neurocognitive assessment, and regarding the imaging, we're doing a T2 flare to look at underlying structural abnormalities, in addition to the functional data that we're going to be getting from a functional MRI. And then laboratory data will be obtained as well. And then 6 months is just a repeat, the neurocognitive assessment will be repeated. And at 1 year and 2 year follow-ups, we're going to repeat those baseline studies. And in terms of functional MRI, for those who are not familiar with functional MRI, so this is, I'm working in a collaboration with the Diagnostic Imaging Science Center, as well as the Integrated Brain Imaging Center, so DISC and IBIC here at UW. They house a 3T MRI system with functional imaging. And when we think about functional MRI, first we're going to be getting a resting functional MRI, a resting fMRI that helps us assess neuronal activity, brain oxygenation. Then we're looking at getting arterial spleen labeling that will allow us to assess cerebral blood flow. And then there's also a breath holding protocol that's part of the assessment. And this allows us to induce sort of a mild hypercapnia and allows us to assess cerebral vascular reactivity. Now, the default mode network is kind of what it sounds like. It's a network in the default mode. These are really brain regions that demonstrate coherent activation patterns and functional connectivity. And so what they were found really when we were, participants were supposed to be at rest while undergoing functional MRI, and were found really to have these areas of the brain that were activated. And so these are thought to be areas that are activated that are associated with internal thoughts, with planning ahead. And so they're activated. They've also been shown to be activated in patients who have depression, anxiety. But when you look at patients with Alzheimer's, there's actually a reduction in brain activity or a disruption in this connectivity. And it's also been found in other diseases that affect cognition as well. So in terms of the neurocognitive assessment tool that I'm going to be using a computerized assessment tool as well as a traditional pencil and paper. And these are the different cognitive domains that are going to be assessed. And these are cognitive domains that have been shown to be affected in sickle cell disease. So a few of them are processing speed, executive functioning, and especially executive functioning is associated with that default mode network that I mentioned earlier. So far, we have assessed about 62 patients for eligibility and have consented 45 participants. And right now we have a total of 10 subjects that have been tested, and I believe one should be getting tested today. So making 11 total. In terms of the data that we do have, so this is for 9 patients. I wasn't able to include the 10th participant and the 11th today. But for the 9 patients that participants that we do have, they comprise of 6 women and 3 men. The age range is about 20 to 55, and most of them have the homozygous S or sickle cell anemia subtype. The average hemoglobin is listed above, and most of them are on hydroxyurea therapy, which is the oldest FDA-approved therapy that we have for sickle cell disease. And one person is on Busola. Busola is another new therapy that helps to stabilize the hemoglobin S and also has been shown to increase hemoglobin levels and decrease hemolysis. And so when we look at the data so far, the images that we have for the participants so far, in terms of the default mode network that I mentioned, we're using a seed-based approach. And the way this works is you have an area of interest that's part of the default mode network, and mostly is the posterior cingulate cortex. And so a seed is placed there, and other areas that have similar connectivity or that are connected to that area should also light up. What we're seeing, this is on the right here, is just one of the examples of one of our participants in the default mode network. But what's notable is that whenever a seed is placed for some of the participants that we're seeing, the other areas are not lighting up. So that functional connectivity is disrupted or affected in some way or delayed. So we're still processing the images, but one thing that was noted is the type of disruption that we're seeing in this connectivity is one that is similar to what would be seen in Moyamoya disease, which is something that is also another unfortunate and common neurovascular complication in sickle cell disease. So it's interesting finding right now. We're still processing the images to see what we end up with, and especially as we get more people participating in the study. And then, as I mentioned, the breath hold protocol, this is one that looks at cerebrovascular reactivity. And so inducing, we're having patients hold their breath to induce hypercapnia. And right now, what we're also seeing is that there's not a good pattern that's observed. So initially it was thought that our patients not holding their breaths correctly. However, I've been in the room and I know that's not the case. But it's also, the other thought is that there could just be low vasodilatory reserve, which has been shown in literature quite a bit in sickle cell disease. And then lastly, when we look at cerebral blood flow, as I mentioned, that's the expectation is that this is supposed to increase and to compensate for anemia and decrease arterial oxygen content. In our group, what we're seeing is lower average cerebral blood flow when you compare to the reported averages in our age group. So we're also looking at the literature. There have been some literature that shows that the way you analyze the data for sickle cell disease when looking at arterial spin labeling, it's a little bit different. So we're also processing the data, keeping this in mind. And then with regards to the neurocognitive data, this is the data by hemoglobin levels. And for the various, except for the executive functioning, all the other cognitive domains, the lower the score, the better the performance. And so when you look here, attention, working memory, and executive functioning, right now there's a trend for those with higher hemoglobin levels performing better. But it's still a small sample size, so I'll be interested to see what the results are when we have a larger sample size. And then when we look at neurocognitive data by age, attention and working memory are also, they seem to be associated with, well, worse performance seems to be associated with older age. Well, I wouldn't say associated given the small sample size, but there's a trend towards worse performance in older age. But as I said before, really, we still have a small sample size. So in terms of the future direction of this study, one, we still have the neurocognitive data to correlate with the different functional MRI data that we have. Hopefully, perhaps we can also find some sort of neuroradiologic marker for cognitive impairment as we process the data. I also would like to extend the study to adolescents. So Seattle's children, we're having a conversation to see potentially could we extend the study over there. And then the other thing would be to have some sort of a comparison study with a control group of either patients who have iron deficiency anemia, because we can compare looking at neurocognitive functioning, looking at imaging at baseline, and then compare to when the iron deficiency is treated and they're restored back to normal hemoglobin values and no longer iron depleted. Do we see the same? Do we see an improvement in cognitive functioning? Do we see similar? What changes do we see on functional imaging? And it will be interesting to compare both groups and see and figure out if there are similar underlying mechanisms for neurocognitive dysfunction. I also would like to eventually hopefully expand the study to other sites. And then one thing that's also of interest for me, right now we're working on resting functional MRI, but a task-based functional MRI is also interesting because you have participants actually undergo cognitive task while under functional MRI, while undergoing the functional MRI, and it will be interesting to see what areas we see are activated or not as activated during these cognitive tasks. And lastly, I'm hoping that this study can lead to clinical trials to determine potential therapeutic interventions. So interventions to either improve or revert if there's some reversible component of cognitive dysfunction, or prevent the development of cognitive functioning. I'm also an advocate for including for a lot of these therapeutic interventional trials that we have now for sickle cell disease, including neurocognitive functioning as a clinical endpoint, because we really just don't know much about it and how we could potentially prevent it. So in terms of my longer-term goals, right now, in addition to this current research, I'm also looking at the impact of hydroxyurea on cognitive functioning in both the pediatric and the adult population. This is a project that's currently that's taking place in Ghana. And then along with additional mentors here, we're also hoping to start a study where we look at N-acetylcysteine and the treatment of acute vaso-occlusive crisis, because we really don't have, other than supportive care, we're limited in treatment in the setting of an acute crisis. And really for my career goal, I just would like to be an expert and a national and global expert in management of sickle cell disease with a focus on improving outcomes and complications in sickle cell disease. And so to conclude, we don't know much about the development of cognitive deficits outside of strokes in this population. The hope is that not the hope, but anemia and decreased brain oxygenation could be a possible underlying mechanism that we hopefully can intervene on. And I'm hoping that this study could lead to potential interventional clinical trials. And so I just would like to give a big thank you to the Department of Medicine and the Diversity Academy Scholarship Award Selection Committee. I'm truly honored for this award. My mentor is Dr. Kleber Fetrin, who is my primary mentor and just has been absolutely wonderful, along with my secondary mentor, Dr. Rebecca Cruz-Jarris. This study is supported by the National Hemophilia Foundation, so I'm grateful for that. And then my collaborative team, the DISC IBIC group, Dr. Rain, Dr. Grabowski, and the staff that's part of the group. I would like to thank our neuropsychologist, Dr. Christopher Rhodes, research coordinators, Zach Gill, and my additional mentors who have been career and research mentors for me, Dr. Apkowitz, Dr. Roscoe, and Dr. Lopez. And last but not the least, I'd like to thank my family. Because of them, I'm doing what I got interested in sickle cell disease, and I've really found a passion for wanting to understand more about the disease. So thank you, everyone, and I am ready to answer any questions.
Thank you, Dr. Oluwole. That's very interesting work and obviously very translational, and it's addressing a major problem that we need better treatments for. So it's certainly an unmet need in medicine. There's one question that there's a couple of questions that came in here, and I'll go with the first one. One audience member asked, what are the RBC transfusion needs of these patients, or the patients in your study? And is there any correlation between transfusion, hemoglobin levels, MRI, imaging, and cognitive data? And in addition to, and then secondarily, they asked, in addition to the hemoglobin levels, are you also evaluating or looking at the percent of hemoglobin S in these patients, and if it correlates with any of these manifestations?
Yeah, so great questions. First question, right now of the participants that we have there, we don't have anybody who is currently on chronic exchange transfusion. The goal is that once we have our sample size, to do some sub-analyses looking at the different treatments that patients are on and seeing if there's any association between what treatment and cognitive performance. Regarding the second question, are there any data regarding I think transfusion, cognitive data, and imaging? So recently, I think it was 2 years ago, there was actually a study that was published that looked at baseline neurocognitive functioning in patients who were treated with chronic transfusion and then tested them a few weeks after transfusion. So a few weeks prior and then a few weeks after transfusion. And what they found was that there was actually an improvement in executive functioning abilities in those participants. The only sort of question, and I think they chose certain tests that would not have any sort of learning associated with it, but there's always concern for learning when you do these cognitive assessments so close. And so that's in terms of imaging data, chronic transfusion has been shown to have an improvement with cerebral blood flow. So that's one thing that we do know. And so when we have more participants who are on red cell exchange, it'll be interesting to also look at other additional fMRI data as well. And there was a last question, and I'm sorry, I think I've forgotten the last question.
The correlation with the levels of sickle hemoglobin and how that then lines up or associates with these manifestations.
Yes. So I presented part of the study at our hematology retreat, and that was one part of this study that I wish I had done, which was including hemoglobin S levels in as part of the study. So patients are not always routinely checked for the hemoglobin S levels. And so to be able to have more participants in the study, I was collecting sort of their routine labs and not adding additional labs for the participants. And so if they had any routine hemoglobin S, it is collected, but I did not, when I initially designed the project, I was not collecting hemoglobin S levels, but it's something that I'm considering including as part of the study.
And also, there's another question I'll bring up here. Comment, great study and excellent presentation. What about the risk mitigation of hyperviscosity that comes with higher hematocrits and sickle cell disease when patients who are transfused? Would you like to comment then on the recommendation to keep the hematocrit at 33 to 36% to avoid hyperviscosity?
Yeah, I think I mean, right now, I don't think I have the, but except for the participants who are on hydroxyurea, we know that their hemoglobin levels do go up. Those on voxelotor, it does go up. But I think for the most part, the higher levels do fall either within or slightly below those recommended values. And so right now, what we're looking at for the higher end is really, it's less than the hematocrit of 12. And so in terms of hyperviscosity is something that we are looking at on imaging to see are we seeing any changes are associated with hyperviscosity. But it's certainly something to be aware of in the future. We're seeing that that higher hemoglobin levels are associated with better cognitive performance, we still have to keep in mind sort of how high is too high for our patients.
And then I'll probably end here with this question, is that you did mention that, the best treatment for sickle cell disease would be to correct the anemia and to correct the sickle cell trait, or the gene. And that's obviously the best approach, but short of that, it seems that some of the pathology is because of sludging in the microvasculature, especially the cerebral vasculature for the cognitive defects. It's akin almost to like happens in cerebral malaria, where you get sort of occlusion of the cerebral vasculature. Besides N-acetylcysteine, are there any strategies that could potentially reduce that sludging in the cerebral vascular system to prevent the neurocognitive defects or deficits that occur in sickle cell disease?
I think one of them is really, right now what we have is the red cell exchange, but as we know, there are complications that come from that, which is, you have iron overload. It's also very inconvenient for participants, but that is one way to potentially prevent sort of the sludging that you would see associated. But it's also higher hematocrit could be associated, so we have to kind of be mindful about what the target hemoglobin levels will be. And there's this current study right now that's ongoing. It's not looking at cerebral vascular disease, but it's looking at pulmonary hypertension, where they're looking at chronic exchange. And so that would also provide a little bit of information about what are the target hemoglobin levels that we should have that could help sort of where we wouldn't see the sludging, but we are able to kind of prevent the sludging.
Mm-hmm. All right. Well, I want to thank both presenters today and offer our congratulations from the Department of Medicine to both Dr. Oluwole as being the DADSA Award winner for 2022, and also to Dr. Li for the Chair of Medicine Scholar Award winner for 2022. Excellent presentations. The Department of Medicine is proud to support your research efforts. And just wanted to mention that these awards come with also some financial awards too. There's $50K per year times 2 years to each of the recipients. And so I'll close and with that, and once again, congratulations, Dr. Oluwole, and also to Dr. Li. Thank you.
And thank you.
And then next week, or not next week, but in 2 weeks, we'll have the Fialco Award winner for 2022 present. And that is going to be Dr. Andrew Sturgakis, and he's going to be talking to us about moving into the unknown, the next stage of genetics and genomics. And he's also from the Division of Medical Genetics. So we'll see you here in 2 weeks. Thank you.